is what the left hand side, what you are going to see, this is what a rubber plant. There it is, you can see there is on a stem or a wood, there we can see there is a mechanical injury with the help of some instruments, and because of the injury, those laticifers are releasing the rubbers, which is again a commercial rubber, which is being going to be collected in this manner. Fine. So just for the point of understanding, I have made this particular one, and you can go for Catharanthus roseus, you can go for euphorbia, you can go for mango, you can go for chico, you can go for any different types of plants. Okay, now broadly, for the point of understanding to you all, one is Apocynaceae, Asclepidaceae, Euphorbiaceae, and Supportaceae. These are the 4 important families that you already come across in your day-to-day life. So these 4 plants comprise latex, milky latex releasing plants. Fine. And fine, again before start of the introduction as well as the different components or different parts of the laticifers, I want to explain where these the location of the laticifers in when you take the transverse section as well as the longitudinal section of a laticifer containing plant. So can you all able to see CO? So this is CO stands for cortex, and this is SS stands for starch sheath. So the can you all able to see the gray colored cells? 1, 2, 3. Gray colored cells are the starch sheath, and the next, can you all able to see this one? Which one? The golden colored circulars. So these are laticifer cells or latex cells. In laticifers, we have different type of latex cells, latex tubes, latex vessels. Okay, now we'll explain you as and when we proceed with the class. And SP stands for secondary phloem, and SX stands for secondary xylem. Don't get confused with that. And in some other plants, this is what before staining. So before staining, what the section, how it is going to look? It looks like this. And after the staining with the different magnificent stains, especially required for the xylem, phloem staining as well as the secondary the laticifer staining, there you can see that SP stands for secondary phloem and SX stands for secondary xylem, that you can see already with the morphological appearance. And these blue colored cells are the latex yielding components or laticifers. So these 2 are the images of the plant which are after taking the transverse section. So here, can you all able to see after taking the longitudinal section, these laticifers are the specialized elongated cells, which usually available in the leaves as well as stems, and they usually considered as latex secreting cells, or they usually secrete latex. One more point is that here I have not mentioned it. Here I have not mentioned even some plants where reproductive parts also may have laticifers. So for knowing that, just go and collect any euphorbia plant, euphorbia genera. Okay, now so there euphorbia genera, just go and pluck the inflorescence or any flower. There you will come to know the release of milky latex. But broadly, most of the plants contains the laticifers in the leaves as well as stems. Very often, I mean, sometimes there are some specialized families, there are specialized what we call genera, and there are some specialized species comprise the laticifers even in the inflorescence or even in the reproductive parts of the plant body. Where the laticifers, the latex secreting structures are of different types in the plant body, and they may be a latex cell, or it may be a latex vessel, or it may be a latex duct, it may be a latex tube, or it may be a laticifer ducts. Nothing but, so we are latex releasing components or latex releasing structures are of different types. They are having different morphological appearance, and their what we call release of latex is also varied. But the chemical components of those latex are again varied in the different latex secreting structures. But here, till now, we have not yet come across, there is no marginal clear-cut information on the differentiable characteristics of the latex cell, latex vessel, latex duct, latex tube, and laticiferous duct. Henceforth, broadly, all these together will be considered as laticifers. Nothing but latex secreting structures. And usually, these laticifers are available or found in approximately, not accurate. I'm not just giving the accurate number with the internet resources. Just what I'm proposing here is that The laticifers are available or found in more than 12,500 species, which are falls under 900 genera, belongs to the 20 families. So to be come out of the plant body as and when there is a mechanical injury, there must be a breakage. Unless until they are the laticiferous will not what we call transfer or secret any of the component outside of the plant body. So people may admit the fact what I'm explaining right now is that, so unless until you are keeping the Vinca rosea plant as it is not going to release anything. But if you go and cut the Vinca rosea plant stem, there you are going to see the milky latex, right? Why such? Because these laticifers are the characteristic feature of these laticifers are they are going to be released. They are secret the latex. It may be in different color, it may be having the different components, but to tell you that in the secretory system, broadly in the plant comprising the secretory system, we have got two types. One is external secretory system, another one is internal secretory system. What is the external? In any mechanical injury without having any damage, they are releasing certain components. So those will be considered as external secretory system. Where internal secretory system only laticifers are the one thing. Okay, now coming to the laticifers, broadly these laticifers are classified into two types. One is articulated laticifers, another one is non-articulated laticifers. Nothing but based on the strata, based on number of cells, based on the arrangement of cells, we have got two types. One is articulated laticifers, another one is non-articulated laticifer cells. What do you mean of articulated laticifer? Nothing but these laticifer cells comprising of many number of cells. Nothing but these cells all together joined one above the other, or one beside with another one, forming a complex structure of a laticifers. Nothing but articulated laticifers are those which comprise a series of n number of cells. Again, the articulation of a laticifers varies from plant to plant. Some plants will be having 10 cells, some plants will be having 100 cells, some plants will be having many number of cells. So the articulation or articulated laticifers are those which contains a series of cells which are joined together. Nothing but one cell is attached to another cell attached to another cell. So it goes on like this. Where non-articulated laticifer cells are those which are only unicelled. Nothing but both articulated laticifers and non-articulated laticifer cells are considered the laticifer cells because they are going to release a huge amount of the latex. It may be in any form. It may be in milky form, or it may be in white liquid form, or it may be in transparent form, it may be in the golden yellow form, it may be in any liquid form. Both the type, articulated or non-articulated laticifers, are going to release the components which is considered as a latex, but the articulated laticifers comprising of many cells which are arranged one end to the another one, where articulated laticifer cells are those very single cell, where these are broadly considered as the xenocytic cells. What is mean of xenocytic cell? Nothing but a cell which is, so usually what happens in a cell division, the karyokinesis occurs and the cytokinesis occurs. Which one occurs first? Whether the karyokinesis or cytokinesis?
Karyokinesis.
Yeah. So here what happens? A cell after undergoing the karyokinesis which fails to undergo cytokinesis. Because of that, a singles are those which comprising of a elongated, big, enlarged cell comprising many nuclei. Nothing but a cell failed to undergo cytokinesis. Right? So here non-articulated laticifer cells are those which are xenocytic cells, or they may be a single celled which containing many nuclei. Laticifers, nothing but latex secreting structures are of different types, as I already told you that. One is a latex cell, latex ducts, or laticiferous ducts, or latex vessel, or a latex tube. As again, I would like to repeat that though we have 4 or 5 different types of latex secreting structures in a plant body, but yet we are not clearly, or marginal distinguishing characteristics are not yet clearly known to us. So all the 4 or all the 5 will be broadly considered as laticifers. Now coming to the latex cell, the first one is that these are the latex cell. They may be a simple latex cell or a branched cell, but usually they are derived from the enlargement of a single cell. A single cell will undergo enlargement and they are going to become what we call the latex cell. However, what happens, these latex cell after the enlargement, they may become branched also, but they never overlap. They may even overlap each other. Nothing but a single cell after enlargement, it may join in different pattern and they may overlap each other, but they never get joined. Nothing but they never get attached to each other. So These are the latex cells. And the next one we have the latex ducts. These are latex ducts are nothing but, I think you might have already heard about the secretory ducts. Similar to that, we are going to see that some of the tissues, they contains the duct-like structures, and they are elongated and branched and aseptate. Nothing but we are going to see that again ducts are elongated structures, again pipe-like structures, and they may have, we are going to see the branches, and they are completely aseptate. Nothing but the septa is completely absent. Now coming to the most important and the what we call the most studied latex secreting structures are the latex vessels. These latex vessels are, they may be a simple or branched tube-like structures, and they usually anastomous. Let me tell you that anastomous nothing but two or many structures are having many openings. Will explain you detail in the image. In the I think you can see the right hand side bottom, right hand bottom side of the image. You can all about these results which are formed as a result of, again, as an enlargement as well as the union of a chain of cells. So these latex vessels are, they may be simple or branched tube-like structure. They are anastomers with similar tubes. Nothing but they what we call the components of a latex vessels will be having a similar structures, and they are formed as a result of the enlargement as well as the union of different latex releasing cells. Next we have latex tubes, and these latex tubes again, this particular latex tube nothing but here we are not yet clearly understand how the latex tube looks like in any plant body, but it has been known from the different research article that they are the modified versions of the latex cell or a latex vessel. Nothing but they comprising one small veins, right? These are the small veins. Fine, let us take this particular vein. This region has got damaged. Then through this it can reach this place, right? Even this portion is damaged. Through this it can reach this region. Nothing but the region or the what we call the components of opening and reaching to a particular positions can be done with a different openings, and those will be considered as anastomos. And the best, actually the anastomos terminology is more employed and more suitable to explain in the kidney in the cells. They are again unbranched here, and you can see that latex vessels. So these are the latex vessels. You can see there are branched one. This one is latex cell, this one is latex vessel, and these two are the images of the tissue after taking a longitudinal section. And when you take a transverse section, here we are going to, can you all able to see the dark black colored structures here? 1, 2, latex vessels. Always remember that the laticifers are always associated with phloem. In the coming slide, will birth. These are the latex duct 1 and 2 images. So here, why I am mentioning the two images of a latex ducts is that, so here this is a latex duct, and this is also a latex duct. Is the lipid ball. Here you can see this one is latex duct. They are devoid of any of the such specialized structures of the latex. I mean the morphological characteristics of a laticifers. Usually the laticifers are the cell wall of the laticifers are quite thick than their adjacent parenchyma cells. But as we already told you that they are associated with the parenchyma cells, I mean the phloem cells. Usually in the previous image you can be able to see these are the parenchyma cells. Okay, so that these laticifers, how to distinguish from the neighboring or adjacent cell is that these are quite thicker than the adjacent parenchyma cells, but the thickening of the cell wall is not because of the lignification. The lignification is not because of the lignification, nothing but they are non-lignified, but their thickening of a cell wall is just because of excess deposition of cellulose, hemicellulose as well as pectin. Nothing but the deposition or the growth of cell wall is occurred by the apposition process. What do you mean by apposition? On the cell wall, that's what the cellulose, hemicellulose and pectin are going to be deposited. That's what we call it as apposition, and they are becoming thick walled than the adjacent parenchyma cells. And the occurrence of a laticifers in some plants what we call the laticifers are found on the throughout the plant body. I mean, they may be available in the leaf, they may be available in the stem, they may be available in the inflorescence or reproductive parts. And in some plants, they are restricted only to the leaf, or some plants they are restricted to the certain tissues only. Where the laticifers are always releases the fluid, nothing but a latex, which is varied in the chemical composition as well as morphologically. Nothing but morphologically, the color of a fluid or a latex varies. In some plants, we are going to see white color latex or milky white color. The example here I mentioned, that is euphorbia, one is asclepias. You can take euphorbia, calotropis, vinca rosea, even the sapota, right? And in some plants, you can see yellow or brown colored latex in the cannabis plant, cannabis sativa. And in some plants, we can see the orange colored or some colorless or the what we call transparent or a clear solution of a latex in the Morus alba plant nodidira. And even in the nerium. Don't get confused the nerium of both primary as well as the secondary metabolites. Nothing but they are having sugars, proteins. Apart from that, they may be having the secondary metabolites like the alkaloids, terpenes, steroids, and they may be having some other components like this oils, mineral salts, organic acids, resins, and the rubber. So these are the different components. I'm giving the general information about the components of a laticifer. But apart from these, also they may be having the different components in the latex, which is going to be released from the laticifers. And here some of the particular example I would like to give and what they contains in the latex. So here Asclepias syrica. Asclepias syrica is a plant name which contains the latex of this particular plant contains pectinase, an enzyme. And the opium, it is again a most medicinally important alkaloid which has been obtained from the papaver. Have you heard the papaver is the latex of the latex contains a rich vitamin B1, where euphorbia milli. So euphorbia important feature is that these star grains are dumbbell in shaped one. Again, one more, that is a natural rubber which has been obtained from the Hevea brasiliensis, which has been scientifically considered called as cis-1,4 polyisoprene, which is a most commercially extracted latex from the rubber plant, again from the stem itself. And this is been used as a commercially important in the what we call in the arms, in the weapons, or it may be in the storage, or it may be in some other what we call industrial components. Only we are going to use the rubber for the rubber as an commercial product from the latex derived from the latex of the Hevea brasiliensis. Again, the vinca contains the latex of vinca contains the different components, the different alkaloids. Don't get confused with vincristin and vinblastin are the two alkaloids. They are available in the latex, though the vinca stem or the plant body of a vinca contains the vincristin as well as the vinblastin, the two components, where the latex of vinca doesn't contain these two components. But there are some other alkaloids are present in the latex of the vinca. Again, one more, the two more important feature of the laticifer containing plants I would like to give here. One is Palacium gutta. So again, this is a plant, botanical name of a plant. They releases gutta percha. Again, this has been conventionally used as after the root canal treatment. Root canal treatment gota hai? The root canal treatment will be made by the latex of Palacium gutta, that is gutta percha. And next is the latex of Acra sapota is chicle. Again, the latex which has been released from the sapota is considered as a chicle, but I mean these chickles are used the chewing gums. So the chewing gums, the latex or the softness of the chewing gum is because of the chicle, that is the latex of the Acra sapota. Can I continue? Ferrous components in the plant body. The component is that how the humans have the blood vessels. These laticifers are also act as a vital sap vessels in the plant body. Nothing but the blood vessels are as vital as for the human or animals. Similarly, the sap vessels, the laticifers are act as a vital sap vessels in the plant body. Nothing but they also act as the store reservoir or store of the food materials, and they may even supply the food material in the young stage. And the translocation of the assimilates nothing but the after the phloem, which has as it is associated with the phloem, how the phloem conducts the food. Similarly, they are undergoing the translocation of some of the assimilates. Nothing but the internal secretory tissue, but the components of the laticiferous or the latex of the plant body will not be utilized by the for the plant metabolism. Nothing but once the latex or once the component which have been synthesized in the laticifers or the structures, they are not going to be reutilized by the plant body. They will remain as it is unless until there is a plant defense mechanism, or unless until there is an what we call the mechanical injury or breakage. These are not going to come out. Once they come out, they will stop the infection caused by the mechanical injury. And apart from that, they are also act as a transporter of the oxygen, as well as they helps in healing up of the wounds. That's already we know that once there is a cut, that to heal that they are going to release some of the latex, and they also acts as a transporter of the oxygen. And apart from that, they are again what we call, they are going to produce a wide array of proteins, and those proteins can be act as a what we call functional enzymes such as pectinases or the chitinases, and these enzymes may act as the defense against the herbivores as well as the microorganisms. So this is what the function of but we have got many possible original or development of the laticifers in a different mannerism or a different pattern. So there is no clear cut. They are going to be released in the particular pattern or from the particular information or a particular initiation. The laticifers are going to be formed. Henceforth, we are not yet clearly understood how they are going to be formed. But there are it is an opinion by the different scientific community that they are polyphyletic in origin. Nothing but the formation which is of when the plant at the embryogenesis stage itself, where the laticifers are going to be formed. Clear laticifers are going to be formed when from the ground meristem as well as the procambium. Am I clear? These of cells, just see they are attaching one to another. There is again gap. Here you can see they are attaching one to each, one above the other, and finally at their maturation stage, you can see there is a single pipe-like structure. There is no septa at all. Here you can see the small septa, small opening, but here there is no septa at all. They are non-branched or non-anastomosing. And here you can see that again a series of cells are joining. Along with that, the neighboring cells are also getting joined and they are becoming branched. Just see, can you all find the difference in this? But a single cell which has got elongated, and they are, let us take broadly, we are considering them as sinocytic cell. You can see the non-articulated, I mean the unbranched laticifers in the initial cells. When the leaf primordia are going to form, you can see that the small laticifers are going to form from the ground meristem as well as procambium, and finally they are going to increase as there is an increase in the plant body cell, and this length or elongation results in the increasing in the laticiferous size. And you can see that non-articulated are branched at the what we call in the initial stage itself. During the embryogenesis itself, you can see the formation of the laticifers, which are very small in size, and you can see at the end very branched, nothing but anastomosing type of the laticifers. Is that clear now?